Hello and welcome, everyone. I am Carmen Guguta, Head of Business Development and Marketing at Technovis Crystallization Systems. I am happy to share with you today that Dr. Sophie Jambon, Team Manager Early Development Phase at AstraZeneca, is joining us today, and she will share her view on what is a scientist's guide to crystallization process development. Before we start, I have a bit of information related to the webinar to share with all of you. So the webinar is being recorded and it will be made available. Tomorrow you will receive an email with a link to the recording. If you have questions, please feel free to write to us making use of the chat function of this webinar. And at the end of the webinar, we will take some questions. I will read the questions to Sophie and we will try to address all your questions. Later on, you can still send us your questions. Write to us at info@crystallizationsystems.com and we will do our best to answer all your questions and we'll forward also your questions to Sophie. The webinar will last about 45 minutes. For those who are not familiar with Technovis Crystallization Systems, we are a Dutch company located in the north of the Netherlands. We are a world-leading technology provider for solid-state research, crystallization, process development, and formulation. We are in business since early 2005. And in our product portfolio, we have currently 3 products, Crystal Reader, Crystal 16, and the Crystalline instruments. Some of you most probably already know the Crystal 16 or the Crystalline or both of these instruments. If after this webinar you think you would like to test one of these instruments, I think the most easy way is to contact us directly via the website or write an email to info@crystallizationsystems.com. We will be happy to further assist you with this. In the meantime, please remember the Crystal 16 is your instrument for solubility, metastable zone with solid state screening, and the Crystalline with the camera technology that works exactly like a microscope, and the possibility to connect it to a Raman spectrometer helps you to develop and optimize your crystallization or formulation process. This was about Technovis Crystallization Systems, and now I'm really happy to introduce to you our speaker today, Dr. Sophie Jambon. Sophie is a French citizen. She moved back in 2004 to the UK when she started her PhD at School of Chemical Engineering and Analytical Sciences at the University of Manchester in the group of the well-known group of Professor Roger Davey. Her research topic was, at that time, understanding crystallization pathways from thermotropic liquid crystalline states. Shortly after, in December 2007, she joined AstraZeneca as senior crystallization scientist, and her activity was to develop a robust crystallization process for APIs to provide suitable crystalline form and particle properties for downstream processes, including efficient isolation, drying, formulation development, and drug product manufacture. In 2017, Sophie was promoted as Associate Principal Scientist in Crystallization Science, and since 2018, she is a team manager of the Early Development Phase group from pre-lead optimization to Phase 2B. She is leading a team of specialists including biocatalysis, crystallization, science, material and particle science, and process engineering. As you can see, Sophie, and I'm pretty sure a lot of you already know Sophie, has a broad experience in crystallization and knowledge, and I cannot be thankful enough, I think, Sophie, for helping us today and bringing today this webinar to our viewers, to our customers, and sharing your view on crystallization process development. I'm making you a presenter now, Sophie. So I think you should be able to share your screen, and the floor is yours. Thank you very much, Sophie.
Thank you very much, Carmen, and good afternoon, everyone. I hope you can see my presentation.
Yes.
Is it now on the presentation mode, Carmen? Just to check.
Yes. Yes, I confirm it's on the presentation mode. What we see.
Perfect. So I'm going to be really honest with all of you. This is my experience I will share with you as a crystallization scientist with over 15 years of experience in the field. And everyone has got different experience, so I hope my experience will help you. And I'm very glad that I got that opportunity from Technobis to actually share my take on crystallization science. And I've give the title of a scientist's guide to crystallization process development because I always like hitchhiker guide to the galaxy. So I thought that would be a nice start of a talk. So I'm going to give you an outline just to make sure that we are all on the same page and we're following that presentation together. So I'm going to give you a very rough introduction to crystallization science. I give you some framework and guidance that we are implementing within AstraZeneca. I give you some example of challenges and then approaches and tools to solve those challenges. I will give you then a conclusion on those on that presentation, and then also maybe some I think food for thought, future perspective to think of when we talk about pharma industry and crystallization science. So first of all, introduction to crystallization science. So what is crystallization science? Okay, I love that picture. I think that 500 years ago, crystallization science or anything which was related to science was basically linked to being witches. Thank God we've moved away from it. And but I love that picture because it's always made me laugh. Sometimes I think that maybe process chemists think I am some kind of witch, but never mind. Aside of that, so basically, crystallization science is not only making crystals. I just want to put that straight. We are focusing on API, so active pharmaceutical ingredient, but we're also involved for intermediates. But our primary focus is definitively the API. And we are basically at the interface between the chemical synthesis of a drug substance and the formulation or drug product. So we are trying to make sure that we are delivering to the drug product the right form, the right particle, the right purity profile, and the right throughput and sustainability. And all those is really to make sure that the patient get the best into the medicines. I think I want to focus a bit more on the form and particle because that's where most of the challenges as a crystallization scientist comes. It's basically in terms of form. I'm going to focus that talk on crystalline, but we also do work with amorphous compound. And in terms of form, is really key to get the right form for manufacturability, manufacturing purposes, but also and for sure, basically making sure that the patient has the right solubility profile coming into when he takes the drug. And then in terms of particle, it's important to think about particle like sugar in the kitchen. You wouldn't use icing sugar to make, or casting sugar or granulated sugar to make the same things. You are going to use those different sugars grades for different things within the baking industry. So it's kind of the same mentality in terms of drug product. So you're sometimes looking for solubility. So you want maybe small particles, and getting basically a better solubility profile, or you might be looking more at flow to go well into the drug product and flow well within the tablet making. So it's this kind of thing we need to think of. So basically, in terms of crystallization processes, we've got a fair example here. We've got, I've highlighted 6 main ones. I put A and B as cooling and anti-solvent because that's the most common one you will find in pharma. That doesn't mean that the others are not used. It's just the common in terms of when you're making free form, most likely. And when I say free form, it's just basically just the API as a crystalline solid. So the cooling will basically, the temperature will control the crystallization. The anti-solvent is basically an anti-solvent added to a solution of the API, which then will generate the crystalline material. And then there is pH change and salt formation, and even also co-crystal formation, in which you will have to add something else into the solution of API to create an environment for the API to crystallize. So pH shift could be just because you have ionized API and you want to turn it into a neutral species, which will crystallize. A salt formation could be a counter-ion you add, and then you form a salt which will crystallize, and then the co-crystal will be you add the co-former, into the solution of API and you form your co-crystal. In terms of distillative and evaporative, it's based on the same method for both, but at different speeds. And it's basically removing the solvent. So by removing the solvent, you're increasing your concentration of your API and then promoting crystallization. So that's basically give you a very quick overview of what is crystallization science. Let's see now how we implement those learnings into our early phase portfolio. So I'm going to show you what we use in our early phase portfolio as a workflow to actually support the development of API process development. It's called the API wall because it's a wall, basically. So the main thing of that is if you don't have the foundation right, then the wall will basically be fragile. So that's kind of this concept. And the main thing to make that wall strong is having a good understanding on your molecular understanding and your form assessment. So understanding what form you're making and what kind of landscape and diversity that form is exhibiting. Then once you know what you're making, then you can basically tailor the process to make the right thing. And then you will start by doing mix to gram in the lab, basically. And then once you basically develop your process and you feel comfortable, you've done it to a certain amount, maybe depending on material availability, could be 10 to 100 grams, then you can go to the manufacturer in drug process development activities, and then you will make more than kilos of material. And then you can really, basically, it's kind of, the proof is in the pudding. Have you managed to develop enough understanding at lab scale to successfully manufacture the API? And then on the right side, you will see there is a risk matrix, which is focusing on the form and particle. And that is basically taking into account the risk you will encounter through the development and manage that risk and to communicate that risk to other stakeholders. So this is really a visual wall, but also enabling basically discussion between different parties. As you are at the interface, as I showed, between drug substance and drug product, you've got many people you need to talk to as a crystallization scientist. So, let's start with the molecular understanding and form assessment. You've seen that that's the foundation of the API wall, and basically if you don't get that foundation, to my mind, I don't know what you need to make. So it's very key to have that foundation right. And the critical activity in that foundation and in that first big brick is basically having an appreciation of the form selection. So is a free form okay? Do you need to go for a salt, or do you need to go for a co-crystal, or do you need to go potentially for an amorphous? So that basically will need to get a good discussion between different parties within the pharmaceutical industry to discuss about the best way to select that form, not only for short term, but also for commercial. Then you will look, once you've decided on the form, so let's say I've decided on a free form, you need to basically define or understand the diversity of that form. Is a free form going to make lots of solvate, hydrates, different hydrous, metastable form, and you need to understand the relationship between them. So that will be a big risk assessment going on there to really select then, to my mind, ideally the more stable anhydrous form of that particular free form. And then, as soon as practically possible, I will want to have a crystal structure data, because that's really give you the information at the heart of the form you're basically going to make, making a new understanding of how the molecules are basically interacting with each other, how the crystal is being packed, and is it a usual or unusual feature you can observe in that crystal structure data. So that's really key to my mind to really start understanding what are you trying to make. So once the form is selected, and usually the most stable form, anhydrous ideally, once the risks are understood, so if you go for an hydrate, you understand what are the conditions to make sure you maintain that hydrate integrity through the entire process and manufacture to the patient. And then once you are when you know what you're trying to make, you can start measuring solubility of the selected form, and that becomes step one. So solubility measurement, I think it's critical as a crystallization scientist to understand your solubility data. So this is a graph of the solubility curve. On x-axis, you've got the temperature. On y-axis, you've got the concentration. And then the dark, the full line is a solubility curve, which will increase usually with temperature. And then above that line, it will be a supersaturated region. And underneath that line, it will be a undersaturated region. And you've got a dashed line, which is basically your metastable zone limit. So this is a dashed line because it's kinetic aspect. So it can change with scale or different input of material quality. So it's important to keep it as a dashed line. And at this point, then you know that nucleation will be spontaneous. So it's really good to understand that basically kinetic aspect and thermodynamic aspect. And then you know exactly when also, ideally, you want to initiate the crystallization by seeding. So you can see that is basically changing with temperature. And it can be quite challenging to measure solubility at different temperatures, unless you can use also the equipment that Technobis that Carmen has presented just earlier, Crystal 16 or Crystalline, which enable you to get that information at elevated temperature. But if you don't have those tools, then isolating the liquors from the slurry when you're doing an equilibrium solubility can be quite challenging. So there is other way to determine solubility at elevated temperature when you've got already data at lower temperature, like 20 or 25, which is much easier to get under normal condition in labs. So you can estimate using the ideal solubility, so the Van 't Hoff equation. It's rough, but can be quite useful. You can use also solubility double every 20 degrees, which is a rule of thumb, and you can find some information in that paper from Franz Müller and all. Or you can also use predictive, but you always need, in terms of predictive, a bit of information in terms of experimental to really get a bit more accurate data. So that's approaches to basically understand and draw that solubility curve. So now I'm going to show you how we use that solubility data on a compound. So I'm going to present you to compound 1, which is a free form. And we've basically measured the solubility of that free form, which is, let's call it for argument's sake here, form A, at 25 degrees in 10 organic solvents. So you can find in x-axis the different solvents, and in the y-axis basically the solubility in mix per mil. And you can see that I've color-coded that graph. So I've color-coded that graph. There seems to be automatic things at the moment, so I need to see why, but never mind. So I've color-coded that slide in terms of green being maybe good for cooling crystallization, amber being maybe more useful for anti-solvent crystallization, and that is only based this assessment on the solubility data. And then red is basically when it's really highly soluble, cannot be used really for cooling crystallization, but could be potentially, if there was no system for cooling crystallization, be used for an anti-solvent crystallization. I want also to say that this traffic light system can also be used to present basically if you have a problem of purity or degradation of the API, also form control. In that case, for me, was so stable in all those solvents, so it was okay. So reading that, it feels like basically propanol and heptane seems to be pretty good for cooling crystallization. So you can see that the solubility at 20 is between 5 and 20 mg per mil. At 25 degrees Celsius, apology. So what we did then, we took those data and we estimated the solubility at elevated temperature using the rule of thumb I introduced to you earlier, which is basically the solubility double every 20 degrees in 2-propanol and heptane. And you can see that the response of solubility in 2-propanol is steeper than in heptane. So we thought maybe 2-propanol might be better. And additional information is also if you have a solubility of 100 mg per ml at 85 degrees Celsius, then in 2-propanol, you could get an estimated yield of 87% when you isolate at 20, or 95% when you isolate at 0 degrees Celsius. So already you've got forecast of what could happen in terms of process outline. So what we did is basically we selected 2-propanol and we did some experiment in the lab, and we get, actually we slightly changed some bits and bobs, but we got a dissolution at 75 degrees Celsius. We initiate the crystallization by seeding in the supersaturated region. We isolate the batch at 0 degrees Celsius and actually got a yield of 90%, which was estimated at 92. And we got a high purity compound. And what was nice as well is the material we make in the lab was basically equivalent to what we make in the pilot plant. So the crystallization behaved exactly like in the lab, and we maintain a high yield and meet specification. So in that case, colloid crystallization was very successful and quite easy to accommodate at larger scale. So basically, I showed you now that by following the workflow, measuring solubility, doing some experiment in the lab, and then going into the pilot plant, then you can see that it's really key to basically go stepwise. What I want to show you now is when things are actually a bit more challenging. So in this case, it was an ideal project. It was my first project within AZ actually, so I was pretty happy and chuffed, and I thought, well, I'm doing really well. But then, hey, new project, new challenges, and that's what I'm going to show you now. So let's talk about compound 2, which is another free form. So the process is this time an anti-solvent process. I do compare those processes as reactive crystallization because, to my mind, you still have this mixing approach when you have to add two liquids together and how they are going to reach an homogeneous state. So that's why I do tend to link them together. So in that process, we basically just going to sorry. In that process, we basically add to a solution of the API an anti-solvent, which then will initiate the crystallization. And we initiate the crystallization by adding seed. So we don't try to make it spontaneous crystallization. And this process works really well up to 20 kilogram batch, and we have successful control of form. But then a scale factor of threefold, and you're thinking threefold is not much. Well, it makes all difference. We might have been on a knife edge, but we generate a mixture of form. So it's like, okay, what's happening here? And how are we going to work on that process to make it working for another 60 kilogram scale? Because otherwise you will have to do 3 times 20 kilogram batch, and that's not really sustainable. So we need to think how to make that work at 60 kilogram. There we go. So we did a cause root analysis approach, and we found out that, yeah, the mixing and the dispersion of the seed to drive the growth of a desired form was actually not optimal, and that's so that's basically increased the risk of making the alternative form. So what we did is we did a bit of prediction using DynoChem. So DynoChem enabled us to calculate the mixing parameter from a large scale and fit it into an Optimax 1 liter vessel. So we basically did a divide by 400-fold the vessel size and try to mimic what we observe on the reactor into a small reactor in the lab. And we did also tailored design of experiment where we look at different agitation. So we did high agitation, 450, 500 RPM. And then we looked at poor agitation with 80 RPM. And then also we looked at low and high seed loading. So we did a very low seed loading of 0.1% with respect to the input and 5% with respect to the input. So it looks like that in a very quick way, this kind of like quick table, which I will be presented in the next slides in a more showing you which experiment I'm talking about. So let's start basically with low seed loading and low mixing, so the yellow box in that design of experiment. And what we did in the Optimax, it's a 1 liter vessel. It's a smart vessel, and you can control the agitation, the temperature. Basically, we're using an overhead stirrer, obviously, to mimic the pilot plant, and we put a PAT probe, which in this case is a Blaze matrix, which is basically an optical microscope within the vessel. And what we did is we run the process with a low mixing at 80 RPM and then a low seed loading at 0.1%. And we observe basically this basically oiling out and phase separation. The crystallization at all did not behave the same way as you observed in the past. And over the weekend, while the entire process should have been completed, basically the material rapidly and fast just came out. And you can see that the vessel was completely covered, and then even the impeller and the temperature probe were covered of material. And what we found out also when we take a sample of that material is basically that this material was not the desired form. So it seems that an oiling out, rapid crystallization, not the wrong form. So let's keep that in mind. Let's go to experiment 2, where with this time you can see the yellow box. We did low seed and high mixing, and this time no visible oiling out. We did see a slow crystallization occurring, and what happened is with time actually the slow crystallization was of a non-desired form, and at the end of the process we actually got a mixture. So when we did this time, the next was high seed and high mixing. So we did 5% of seed, and then this time 450, 500 RPM mixing. Again, no visible oiling out, all good, but the crystallization was comparable to previous manufacture, the one which were run up to 20 kilogram. And we only got the desired form. So it's like, okay, interesting. So the seed and the mixing together, it's the way forward. So we decided to recommend for the 60 kilogram to actually really add 5% of seed. So increase the seeding. And it's clear from that experiment that the mixing at 60 kilogram was not equivalent at 20. Another thing is we did not actually do the last experiment. We had enough from the first experiment to really come up with a recommendation for the pilot plant and the next manufacturer. With this increase of seed, we actually promote crystallize only the desired form. And then I just want to say that the PAT approach was very highly insightful in that and reduced really our time in terms of understanding process. So that's pretty much a great story for compound 2. So what was the problem, what we came up with, and how we managed to go around and basically successfully deliver a single free form, single batch, and pure form. So I have presented 2 simple. Oh my God, sorry, that should not have happened. So here is basically compound 3. So compound 3 is basically an intermediate this time. So it's 2 batches of approximately 15 kilogram each. It's an anhydrous free form again. And just what I want to say is this time the 2 batches behave differently during the filtration and the drying on the pressure filter. So the pressure filter, it's pictured with a pink drawing on your right bottom of your screen. And you can see that the cake is in the darker color. The pressure comes from the big arrow on the top, and then the liquor is basically displaced from the bottom. And those 2 batches basically behave completely differently because one isolated and dry in 2 days when the other one isolated and dry in 2 weeks. So a world apart. And it's trying to understand what happened there, because in terms of batch cycling time, it's very hard to forecast a delivery when you've got so much variability. So first thing first, as a crystallization scientist, I did basically I've done basically optical microscope. So you can see the two batches. Batch 1 is like large chunk crystals, and then batch 2 is like thin needles. So you can clearly from there understand why batch 1 took 2 days and batch 2 took 2 weeks. And then by XRPD, first thing to be done. Oh my God, I don't know what happened. Sorry, guys. And by XRPD, you can see that it's 2 different forms. So the XRPD are very different from each other. So it's 2 different forms. And then we did additional chemical analysis, and it appears from all over chemical analysis that it was 2 true polymorph, 2 anhydrous form. So what to do then from there? So to understand which form is a more stable form in the current process, to make sure we can actually control a more thermodynamic stable form. That was the first thing which came up to my mind. So we decided to do solubility measurement of batch 1 and batch 2. So basically we took batch 1 and we took batch 2, and we measure the solubility in those different solvents. So you've got only water, aqueous DMSO, aqueous acetone, aqueous acetonitrile, aqueous DMSO, and then anisole. And we measure the solubility at 25 degrees Celsius, and then we do the ratio of the solubility between batch 1 and batch 2. And clearly that the ratio of solubility is going from 10 to less than 1 by just changing somehow the water content. So that kind of highlights something, which means that that's not true polymorph, in the sense that they are not the same chemical entities. Something else is there, and water is playing a role. So that's what we realized is actually batch 1 was a channel hydrate, a non-stoichiometric channel hydrate, when batch 2 was actually an anhydrous form. So knowing that, and knowing also the next step in the chemistry, we understood that form selection for that step was actually something to discuss and agree, and not only around the stability, but also around what will be the next step in the chemistry. And actually, when you look at the next step in chemistry, which did not like any water, we had to deliver the anhydrous form. And luckily, in the current process, the anhydrous form was a more stable form. So we decided to basically work on the basically, I have to rethink now, work on the seeding protocol. So what we did is we looked at the process, and we decided to see where we could actually seed the process to promote growth of particles which are related to batch 2, and basically enable and promote a better crystallization control. And that was really successful, because we managed to form only batches making only the batch 2 form, but also managed to deliver 3 batches in less than 2 weeks, which is a lot better than the 1 batch in 2 weeks. So basically, understanding your form, understanding your process, link both together, and then come up with a good seeding protocol, then that's enable a great control and a much better process. So that was example compound 3. I'm going straight to compound 5. I was speaking about compound 4, but I know I can waffle for ages, so I prefer focusing on compound 5. So compound 5 is basically a salt, and is made from a salt formation. It's a reactive crystallization process. It was done in production 1 as a primarily single particles, and the particle size of that batch was around, as a D90, around 50-55 microns. And then actually production 2 will actually deliver a larger material because they were bigger than what was made for clinical. And the D90 was around 75 microns. And the difference between both was basically this formation of agglomerates. So you can see those agglomerates sticking. So the larger surface you can see, which is on your front of the picture, basically all the crystals are sticking on that crystal face. So by preferred orientation, we managed to locate where that face is, and it's where you have basically the sodium and the water. So this API, this compound 5 is made of API, but also 2 atoms of sodium and 6 molecules of water. And on that particular phase, the water and the sodium are located. So we were thinking, using crystal structure, can we understand how the agglomeration could occur, and mechanistic, and the rationale to why. So let's go a bit into structural informatics for a little while. So here is the 6 molecules of water in that API. And you can see I describe in the right side the different environment. And also it's something to bear in mind, obviously water is part of the process solvent, because you need water to make sure that the hydrate is stable in that environment. You have also to note, I also want to show you that all these water molecules appear really tightly and optimally bonded to their neighboring molecules as coordination bonds with the sodium, or as edge bonds with the API or other water molecule. What I want to highlight to you now is basically using the Cambridge structural database to give you an insight of usual or unusual of crystal features. So in the CSD, which is a Cambridge structural database, and that has been highlighted in a meeting in 2018 by CCDC, the Cambridge Crystallography Data Center, that hydrates tend at 20%, 21%, and 49% to be donating twice, and 21% to be donating twice and accepting once, and 49% to be donating twice and accepting twice. So that's what means the D is a donor and the A is acceptors. And you can see that basically all these water molecules meet that criteria. They all donate twice and at least accept once. So it seems that the water is really optimally bonding with this crystal environment. So we were thinking, when we saw that, maybe we should reduce the amount of water to reduce potentially the stickiness and reduce the agglomeration. We did not have a possibility to test that hypothesis, but in this case, I just wanted to show you an example of how you could use crystal structure data to inform and guide your experimental work. Let's look now at the sodium atoms. So we've got 2 sodium atoms, and sodium tend to actually optimally more coordinate 6 times, like in sodium chloride, an octahedral system. And in this one, you can see that sodium 1 is only bonded, sodium 2, sorry, is, it seems to be a problem. Yeah, never mind. One of them is bonded only, yeah, just one of them should actually be bonded 5 times, not both of them. Sorry, I must have made a mistake there. But one of them is definitely bonded just 5 times compared to the normal optimal known system. So we were thinking, if that sodium is at this surface and is not optimally bonded, then is actually want to bond further to find that 6th friend. And that might make that particular phase even stickier than normal. So that is actually most likely the reason to why these crystals are sticking to each other. And that gives you a bit of a rational, that's the hypothesis at the moment we are working on. So I give you a bit of a crystal structure aspect and rationale to explain and understand what's going on in your batch, but we still have to have the understanding on what we are going to do with the batch to make it a success. So we need to remind ourselves that it's a reactive crystallization, that we put to clinical phases most likely and primarily single particles with a D90 up to 55 microns. Now we are generating agglomerates, which are increasing our particle size to 75 microns. So we still have next campaign. We still need to deliver for our patients, and we still need to understand what can we and can't we do. So a lot of work going in the background, but we decided us to focus on basically forecasting what could happen for production number 3. So just to give you a bit of, I think, showing you a bit the challenge, okay? So your x-axis here is basically the scale in liters, and your y-axis is basically the risk based on particle size data. You can see that P1 and P2 are the same scale. They are 330 liters. You can see also that P1 and P2 do not share the same impeller configuration. So that's very important to highlight. And I put P1 below P2 because the particle size in P2 were much higher. Okay, so that is basically where we were at. Now the question and the challenge is when we go to P3, and you can see that the scale is 6-fold this time. Remember compound 2, just 3-fold, and we were encountering challenges, so that is 6-fold. And you can see the impeller is different as well. So now it's basically understanding where P3 is going to be. Is it going to be with our clinical plan, or is it going to be outside of clinical plan? So I think that's that kind of thing I want you to really grasp and the challenge we were encountering. So what we did is we did a mix of experimental work at 20-liter scale, which was a scale down of P3, and we call it L3 for lab 3. We did some prediction work, so we did some dynochem calculation for all setups, and we also did some computational fluid dynamic simulation for all setups as well. So all that combined, we came up with some clear result, clear steer on what could happen for P3 basically. So first of all, the shear rate. So the shear rate is the movement of the layers of liquid in the batch, which are going to basically be being moved about by the impeller. And the higher the shear rate, the more agitation and the more layers of liquid basically being agitated, and therefore promoting more particle collision, and therefore enhancing the agglomeration and attrition. So in that, we found out that actually P2 has a much higher shear rate than P1. And actually, we forecasted that P3 should be located between P2 and P1. So it's like, it seems we are still going to make agglomerates for P3. And when we looked at the mesomixing scale time, so basically, it's the time which relates to how long it takes for a drop of liquid to reach homogeneous state when it's added to another to the batch. So basically, the higher the scale, the longer it will take to reach an homogeneous state. And that is basically presenting by that, because in the 20-liter L3, you will have it will be 5 times faster than in P3 to reach homogeneous state. So L3 is not actually perfectly mimicking P3, just because the speed at which you reach an homogeneous state when you're adding your salt forming agent. So you can believe now that if your crystallization is as fast as in L3 the homogeneous state to be reached, then you are in a control zone. But if it's faster than the time you reach your homogeneous state, you're basically going to be in an environment you haven't controlled, and you have a large range of supersaturation spread in your entire batch, and higher presence of hotspots in which you have high supersaturation and lower supersaturation. So you're going to crystallize in an environment you're not controlling. So, as I said for compound 5, basically crystallization kinetics is expected to be much faster than actually the mixing efficiency at P3 scale compared to L3. So we are forecasting that P3 will make large level of agglomerates. And we have actually had the opportunity to implement different scenarios for P3. So we made, I can't remember, but more than 3 batches with 3 different scenarios with different fill levels. So basically the level of liquid at the start of the batch, and then different agitation speed. And we realized that so you can see the particle size here. So L3 was a lot more monomodal. When you go to P3C and P3A, you got bimodal, and then P3B is slightly with a shoulder. So actually, the one which shows the less, the more monomodal approach for P3 is P3B, which relates to a high fill level but a low agitation speed. When P3A relate to a low field level and a high agitation speed, and P3C relate to high field level and fast agitation speed. So it seems that a low agitation is actually the best. So somehow mixing efficiency may not be fast mixing. It can be also low mixing, understanding in this case that you try to reduce particle collision. So I wanted to show you on compound 5 that we've used different approaches and tools. First, to have basically the fundamental understanding to why agglomeration could occur for that compound. And we look basically at the water and the sodium. So it does really help and guide the experimental work. And then we use also a predictive approach together with experimental work to really understand how we can, practically speaking, reduce the risk of agglomerates and control the particle size. And what I said to you, basically, in this case, fast mixing was not the right approach. Reducing the particle collision was the best approach, so reducing the agitation speed. So that's basically the conclusion of the challenging approaches and tools I wanted to share with you in terms of crystallization science. So I hope I show you that crystallization science we observe many different challenges, and they often open at scale up because it's where it's more critical, it's when you need really the material for the patient. And then it's also something to bear in mind is not only the scale, but equipment can also have an effect in terms of pressure filter or centrifuge for the isolation step, for example. So it's really important to not think when you scale up just about the crystallizer, but all the unit operation until you get the API in the bag. I think I show you, I hope, how we can use PAT probes to identify causes of problems in crystallization. And I show you that through compound 2. I show you also that seeding protocol, and really developing a seeding protocol is key to aid crystallization control. And I hope I show you not only that for compound 2, but also for compound 3. I didn't show you compound 4, but it was also another example to compound 5 to show you that crystal structure data really gives you a bit more fundamental understanding and guide the development work. And then I hope I gave you a bit of an appreciation that predictive approach is a very key tool to facilitate decision making for, especially in those cases, scale up. And that I show you for compound 2 and 5 using DynoChem and computational field dynamic. So to finalize the conclusion, I hope I give you a good overview of what crystallization science is and a framework and a guideline to help you to decide how you basically develop a process, and especially know as much as practically possible in the lab before you scaling the process up. I will manage to speak English properly. I just want to make sure that you understand that challenges always can happen during development, especially on scale. And I hope I give you a bit of an overview of some example of tools and approaches you can use in the lab and at scale to basically get better control. Now I would like to give you a bit of a future perspective and also food for thoughts. So first of all, what I would like to tell you is basically within AZ, I've realized that the complexity of our molecule is changing rapidly. We are getting more complex molecule, and actually more complex molecule means we've got limited data of around some key moieties. So we tend to use the Cambridge structural database, which contains more than 1 million crystal structure, to get us some information about things and to predict things. And when you've got exotic moieties which are new, then it's really hard to actually forecast using prior knowledge. We also encountering molecules which are larger at the moment, so like PROTACs with molar mass well above 1,000. So it's also something larger molecular weight, therefore more flexibility and often I've seen and observed a slower crystallization. And I think it's basically right at this moment, really things are changing at a fast pace. I mean, we've seen with COVID, people got used to a different new way of working very rapidly. And is basically, is our knowledge right now ready? And I think we need to really be ready, be prepared, because it's going to be a fabulous, great opportunity to learn a lot about new things. And I'm going to show you what are the new things. So just to come back to the databases, which I touched upon, I just want to show you that we've got already some discrepancies in some ways in some in different databases and data sets between the Cambridge structural data set, the Cambridge structural drug subset, Pfizer and AstraZeneca, and you can read a bit more in that paper from Bryant and al. From 2019. And this paper is really good because it's really showing the differences between those different databases, and the fact that pharma seems to have larger molecular weight structure, that it seems that we've got larger difference between acceptor and donor at AstraZeneca compared to the other database, and also because of more larger molecular weight, obviously more rotatable bonds for pharma. So you can see that there is already differences between databases, and this chemical diversity between databases is already present. So in some ways we need to upskill our CSD to be a lot more relevant for our industry. And I'm not talking only about pharma, obviously. And that's, I mean, CCDC is doing a fantastic job in that. And I love working with them. So I must admit they are really at the forefront for that. In terms of new molecules, oh my God, look at that. I mean, we are not in lack of new opportunities to learn and new challenges. I did lots of my experiences on small molecule, but we've got PROTACs, we've got oligonucleotide, we've got peptide, protein, and all this wonderful molecule coming to our pipeline. It's great for the patient, and it's great as a scientist to be always challenged and learn more, but there are plenty of things to be doing, I can tell you. Also, something I wanted to share with you is the fact that our success rate from candidate drug nomination to phase 3 has increased by basically almost fourfold from 2005 to 2010, from 4% 2012 2016 to 19%. And you can see more on that from that paper from Morgan and all, with the information provided below. So basically, there is an aspiration to really reduce the time of development to make sure that these invaluable drugs are basically reaching the patient. I mean, we've seen some tremendous drug within our portfolio, which in 3 years' time reach the patient, like Tagrisso. But COVID-19 vaccination has also been an amazing example, and not only for AstraZeneca. You've seen other companies doing on the same page. When people are putting their effort, it's fantastic. But then it means it's pressure for us. What next? So we have to basically be really innovative in our approach, and that's what makes my job so great. I think my final thought is, because the time of drug development is going it's an aspiration to be faster, and to make sure that the important drug arrive to patients, we need to be even better to the way we develop processes faster. So I've showed you that cooling crystallization processes can be easier, not easy, easier to develop. And you can see that is basically my take. I'm going to be clear here, my take on what I've observed in my experience around basically different crystallization processes. So if you see the X scale is on liter, and the x-axis, sorry, is on liters, and the y-axis is basically the effort and the amount of work you have to put in. And you can see that cooling crystallization tend to be easier, not easy, compared to anti-solvent and reactive crystallization. So you need to put a lot more effort in those to make them work in batch mode. But we've got plenty of work ongoing at the moment, actually, on continuous crystallization to actually reduce that work and effort, because the scale is not there anymore, and it's just in relation to time. And we involve AstraZeneca with CMAC on that, because we believe that will be a great step forward. So I think what I want to do now is finish the talk, acknowledge all my work colleagues. I've learned so much with them for the past 12 years, I think. I mean, and also during my PhD. So I've worked with them on many projects in the past, present, and future, and I look forward for the future because it's just going to be so cool and so challenging. I want to thank the Reverend Richie Guillon for his picture of witches. He did it himself. Great, I love that. And I want to thank you all for listening to me today. And I hope I was not too boring, and it was enjoyable for everyone, and you've learned something. And I'm happy to get any question.
Thank you very much, Sophie. It was really a great presentation. We have a few questions. As always, a pleasure to listen to your talks and always learning something from you. Thank you very much. We have a few questions.
I took an hour. Oh my goodness.
we enjoyed it, and most of the people stayed online up to now that you were saying goodbye, so that's always a good thing. The questions, let's one first question is from Mark Jackson, and he's asking, when crystallizing compound 1, the material was heated to 75 degrees and sealed to at 65. Was the supersaturation measured at this point? Do you recommend a general percentage supersaturation for a starting point when looking at seeding crystallization?
I think it's case by case. I'm going to be really honest because to my mind and to my experience, every API has got different kinetics. So I'd say in that case, the clearing point was observed at 75, and that's why we seeded at 65, 10 degrees below. Now it's depending on which growth or nucleation aspect you want to try to control, and it's where you are going to decide where to seed. I would not say there is a particular supersaturation which is the ideal point to my mind, from my experience. I think I would just, it's a trial and error, and you need to understand with your path probes, such as a particle tracking probe, basically, where you're teasing more the growth of the nucleation. That is depending exactly what you want. So it's not really a black and white answers, if that makes sense.
Yes. For compound 3, did you manage to change the morphology of batch 2 polymorph? And if yes, how?
So the morphology in terms of particle.
Yes.
Because
yes.
Because the main thing at the time, it was to control the form. So our main interest was really to control the form. I think the form was the criticality there. So the particle we had to evaluate the particle properties afterward, unfortunately. So that was an additional piece of work that we had to undertake after crystallizing the API. So we always have risk assessment, and we all agree that the priority was the form. So that's why we decided to just focus on the form and then do the after work to make sure that we will give the right thing to the patient later. We did not, did that make sense?
Yes. What do you think? How the crystallization process can be achieved for big flexible molecules like Protex?
Oh, slow. Basically, I did a presentation a while back to another group of scientists, and I think I did say crystallization is a matter of patience. Yes, be patient, especially when you have floppy molecules. It's really important to be patient. I'm going to be honest here, I haven't worked yet with a Protac molecule. So, but I've worked with molecules which are close to that same flexibility, and I've realized that you need time for the molecule to basically get the right conformation and then crystallize. But it is a trend, not a general rule for everything. It's really important to understand your molecule. Each molecule is a different case. You can't put them in the same basket. I'm going to be clear here. It's always to try and understand your molecule, and the kinetics of each molecule is different.
Completely agree there, Sophie. I always have said to my team and to the customers I have worked with in the past, get to know first really well your molecule, and you cannot generalize. Each molecule is like an individual, is different. You need to, you can use some general guidelines and of course science, but still each compound will teach you something else. Every time you learn something else, you need to do something else to fix an issue. That's all.
Yep, correct.
We have quite a few other questions. Thanks for the detailed presentation. You have received many thanks. Great presentation. A lot of good wishes, Sophie. I'm happy to share that with you. One question is, I was wondering whether, in your experience, the crystallization of zwitterions had any specificity. Sorry. Here English doesn't help me, my pronunciation. Any specificities that need to be considered? So any specific things that you need to consider or have in mind when working with zwitterions?
I would say first and foremost, it's basically, so first and foremost, when I've got zwitterionic molecule, I tend to go to one of my work colleagues who's got a very good background on PKs, and I go to her and I speak to her, and she gave me like a speciation diagram. So for me, the speciation diagram is key to understand what you're actually trying to crystallize, because zwitterion is maybe in a certain pH range, so you really need to understand which pH range you're trying to crystallize your compound. It's not my area of expertise, because in crystallization science, you've got lots of different, I would say, even more specialized parts. So we tend to work together as a group as well. It's very key on that. Everyone has got their little specialty, if that makes sense. But I would say first, understanding your pKa profile and your pH profile and the species you're trying to make.
Another question of one of the first actually questions I missed to read to you was somebody was asking how to approach low melting crystallization material, so a low melting API. Well, I think we have to get to know your compound as soon as possible, as early as possible, right?
Yes, exactly. I think understand DSC, TGA, solid state characterization should be done even before trying to crystallize a compound. But obviously, I'm saying that sometimes you have to make it first to characterize it. So it's basically a chicken and an egg in some way. I think if you know that it's a low melting compound, it's dependent on which temperature will melt, and it's maybe thinking more doing an anti-solvent at room temperature, for example. That might be one. But it's an approach and an example I'm giving. I think it's really go back to understanding what you're trying to make and in which temperature profile you should be making your compound. So that is part of your, as I was saying, your API role, the foundation, understanding what you are trying to make, what are the physical, chemical attributes of the specific form you're trying to do, to tailor your process condition around that.
Yes. And of course, if it is a possibility, you can also modify by making a salt co-crystal on the melting point of your API.
No, that I completely, I forgot to mention that, and that's a very good point, Carmen. I forgot that, but you're completely right. Yes, there is also alternative to do alternative forms. Agree. Yes. I'm always trying to think, you give me that form. Yeah, trying to crystallize it. It's like, step back. You can do something else.
Yes. One more question I will take. Again, thank you. Really good presentation, great approach. What role secondary nucleation played when you found high speed and high loading gave the desired form? High loading and high speed would have given a lot more seed nuclei of the desired form.
Would it be, I mean, because of the time and everyone else, I think, would it be possible maybe to take those questions?
Yeah, I will send to you this the email, and then you can answer further this question related to the seeding and the speed, the mixing. Indeed. We have actually answered most of the questions, Sophie. Once again, thank you very much. It was really pleasant to hear you talking. And like always, like I said, we always learn. I always learn something from you. For those that they are still online and can hear us this summer, this is our last webinar for the summer summer vacation starts. Already sun is shining in the Netherlands. We are enjoying it. Nevertheless, we are back in September, 16 September, with our first event, online workshop. We are this year celebrating 16 years of the Crystal 16. So 16 September this year, after the summer, we are celebrating, we are starting our celebration of the Crystal 16 with a workshop, a half-day workshop. We will start midday to be able to catch up also a bit of US and still a bit of Asia, as our customers are worldwide. I can already confirm a few of our future presenters at the workshop. Our first user, Dr. Ivan Marciano from Pfizer, Professor Job ter Horst, that now moved to University of Rouen, and quite a few others are on the agenda. This is what I remember at this moment. You will see our campaigns coming over end of the summer in August. We will start to promote this. Once again, Sophie, thank you very much. Thank you to all our attendees today that they have joined us for this webinar. I wish you all a great summer. Stay healthy, and hope to see you all back in September for our celebrating together with us 16 years of Crystal 16. Bye.
Thank you very much, Carmen. Bye, everyone.